Well, good afternoon. My name is Nick Byrne, and I'd like to welcome you to today's ENS virtual seminar symposium, Novel Mechanisms of ENS Dysfunction, Neuromuscular Dysfunction, and GI Disorders. Before we get started this afternoon, I'd like to just give you a couple of reminders. First, October 14th is the deadline for the 2022 grant opportunities. This includes the ENS Discovery Grants and the Transition Award. And mark your calendar for August 11th through the 13th of 2023, as that's the date of our next ENS annual meeting in Austin, Texas. Now, it's a pleasure to introduce this afternoon our two moderators. First, Dr. Elizabeth Videlock, who is Assistant Professor of Medicine in the Division of Digestive Diseases at UCLA. She's an early stage investigator with an interest in the role of intestinal mitochondrial dysfunction and the pathogenesis of Parkinson's disease. Our other moderator this afternoon is Dr. Tamas Ordog, who is Professor of Physiology and Director of Gastroenterology Research Unit in the Division of Gastroenterology and Hepatology at the Mayo Clinic in Rochester, Minnesota. He is also Genetics and Epigenetics Mechanistic Research Theme Group Director and Epigenetics Core Director in the Center for Cell Signaling and Gastroenterology. His research focuses on the transcriptional, epigenetic, and metabolic regulation of gene expression in interstitial cells of Cajal, gastrointestinal tumors, and enteric neurons. And with that, I will turn the program over to our moderators.
Good evening. It is my great pleasure to introduce today's first speaker, Dr. Shanthi Srinivasan. She's actually so prominent in our scientific community that, as moderators often say, she needs no introduction. But for the benefit of those that are new to GI motility research, Dr. Srinivasan is Professor of Medicine and Director of the Division of Digestive Diseases at Emory University in Atlanta, Georgia. Her research interests focus on the enteric nervous system and mechanisms underlying gastrointestinal motility disorders. She also studies the role of neurotrophic factors in regulating hepatic steatosis. Dr. Srinivasan's lab is well funded through the NIH and the United States Department of Veterans Affairs. She was recently awarded the VA Senior Clinician Investigator Scientist Award, which is a major accomplishment. She has more than 75 manuscripts published, some of which appeared in top journals like Gastroenterology, JCI, and the FASEB journal. Dr. Srinivasan also performs a lot of service to our scientific community. She's a counselor for the American Neurogastroenterology and Motility Society and vice chair of the Neurogastroenterology and Motility section of the AGA Institute Council. She's also accomplished as a mentor to students and junior faculty. Today, Dr. Srinivasan will talk about novel mechanisms for enteric neuronal degeneration and gastrointestinal motility disorders. Before I hand it over to her, I would like to remind our audience to enter questions into the Q&A box and not into the chat. Okay? Questions will be answered at the end of the webinar or on Doc Matter should we run out of time. And with this, Shanthi, the floor is yours.
So thank you, Tamas, for that very kind introduction. I also want to thank ENS for this opportunity to present some work on ENS neurodegeneration over the next 20 minutes. It's wonderful to be here and a true honor to be here. So I'm going to share my screen. Let's see here. So today I'm going to talk to you about some novel mechanisms for ENS neurodegeneration. And just to set the stage, as all of us know, when we think about the pathophysiology of motility disorders, we know there's the involvement of the brain, the extrinsic innervation of the GI tract, both the parasympathetic and sympathetic innervation, as well as the enteric nervous system. There can be abnormalities in any of these organs, as we know. And as shown in the cartoon, this figure is taken from Cara Margolis's recent review in Gastroenterology, a very nice review in which we see that besides the enteric nervous system, within the GI tract, there's so much interaction between microbial metabolites, the entire gut microbiome, interacting with the enteric nervous system, the enteroendocrine cells, the glial cells, interstitial cells of Cajal. And we're going to hear more about this from Brian in the next talk. But today, my focus is going to be on the enteric nervous system. And as shown on this slide, as I just mentioned, gastrointestinal motility disorders could be secondary to immune dysfunction, changes in GI secretion, altered gut motility, secondary to dysbiosis, changes in bile acid secretion, and of course, the brain-gut axis, changes in the brain-gut axis. My focus today is going to be, as I mentioned, on the ENS, and this figure taken from Meena Rao's review in Nature Reviews shows just a basic depiction of the enteric nervous system, but just to highlight that the ENS has motor function, as we all know, regulating peristalsis of the intestine, as well as sensory regulation, as well as control of secretion. And we all know it's subdivided into the submucosal plexus lying just below the mucosal lining of the intestine, and the myenteric plexus, which is sandwiched between the muscular layers. So over the years, so I have been studying the enteric nervous system over the greater than past 15 years or so. And we've noted changes in the enteric nervous system in models of diabetes, in Parkinson's, more recently with Dr. Anand Jain at Emory has demonstrated some evidence of kind of mechanism of loss of nitrogen neurons in achalasia. There have been changes in the ENS in irritable bowel syndrome, as well as the ENS has been a target for, in multiple sclerosis. So today I'm going to focus more on our diabetes models and high-fat diet-induced, glucose intolerance, and so high-fat diet models. And so, as mentioned in this slide today, I want to show you some of the changes that we've previously published in diabetic models, as well as the role of the gut microbiome. But most of the talk, I'm going to focus on the high-fat diet model, where feeding a mouse with a high-fat diet, or what we're calling a Western diet, we believe leads to chronic, it's a state of chronic inflammation, leading to oxidative stress and ultimately enteric neuronal damage. So I want to start back where we first published in 2006 in JCI. This was work done by Anita in my lab, and I just want to depict this slide where where we used a streptozotocin model of diabetes, and after inducing diabetes in mice, after 8 weeks, we noted under electron microscopy there were changes in as seen here in the lower panel, the mitochondria within the myenteric ganglia and in the enteric neurons. There was swelling of the mitochondria, evidence of mitochondrial dysfunction. And I show this slide because as I come to our more recent data, we're again focusing on the mitochondria and changes in mitochondrial dysfunction. So I'll bring you back to, how in different models we're finding a role of the mitochondrial dysfunction really contributing to enteric neuropathy. And on the right here, I just want to, I've included some of our earlier work where we showed the PI3 kinase GSK3 beta pathway was important in neuronal survival. So this was in our diabetes model. We also did some studies in human sections from colons of diabetic patients and appropriate controls. This was surgical specimens from patients who had had colectomy due to polyps. And what we found was that the myenteric ganglion area was significantly lower in diabetic patients compared to controls, indicating that there is some evidence of neurodegeneration and neuronal loss. And I know several others have demonstrated this in diabetes. Today, I'm actually, due to the limitation of time, I'm just going to highlight a very few studies. And so I just wanted to mention that. Now, as many of you know, we have a system to study the enteric nervous system. It's an enteric neuronal cell line that we developed in our lab and published in 2008 in gastroenterology. And I've included this slide as many of us have used this as a model to study the ENS. However, I also want to mention that we complement all our work with doing similar studies, not only in the cell line, but also in primary enteric neuron culture isolated from mouse intestines. But this is a model that has been available to us, and I will show you some data from this model also. I have two slides just on the ENS and gut microbiome, experiments that we did over the years, where we showed the role of TLR4. As you know, TLR4 is a pathogen recognition receptor. And in this study that published in gastroenterology, we demonstrated that if in a TLR4 knockout mice, there's loss of nitrogen neurons. And as seen in the top here, top panel, there's loss of nitrogen neurons. And if on the right here, in a study looking at, intestinal motility or transit across the small intestine and colon, there was a shift to the left in the TLR4 knockout mice, indicating slower motility. And so what this shows is when there are less nitrogen neurons in this TLR4 knockout mouse, there was delayed motility. And this indicated to us the importance of the gut microbiome in the development and formation of the enteric neurons. And I know several others have gone on to publish the importance of gut microbiota in modulating both ENS development as well as ENS function. So just moving on to, why we then moved to a different model after diabetes and trying to understand, we decided to study a model involving high-fat diet. And just to set the stage of why we picked this model, we also looked in human data to see if high-fat diet can affect GI motility. And in this particular study, which was done using the NHANES database, we analyzed about 6,000 data from 6,000 patients. And just to highlight the key finding, what we found in these patients where there was a detailed diet, available for us to probe, with a high-fat diet, there was evidence of constipation, a higher odds ratio, a significantly higher odds ratio in people on a high-fat diet, as well as in people and in patients with diabetes, whether uncontrolled diabetes, the constipation was significantly higher and also higher odds ratio in controlled diabetes. On a high-fat diet. So we did this study and we were able to find evidence that a high-fat diet can lead to motility changes, particularly in this case, constipation. And as I mentioned before, we believe it's the low-grade inflammation that can lead to several consequences and ultimately neuronal damage, as I'll show you on the next several slides. So this study, we tried to examine the effect of a high-fat diet focusing on high saturated fats. I'm just showing one figure from this study where we found evidence of apoptosis with high levels of palmitate in enteric neurons. And this was the first study also where we started to look at evidence of changes in mitochondrial function. And I'll show that further in subsequent slides. We also, what I mentioned the role of gut microbiome and role of TLR4. When we started our studies with high-fat diet, we were able to find there was, as is known, there was a gut microbial dysbiosis. And as shown on this top panel, there is evidence of change in the gut microbiome with the high-fat diet. I should mention our high-fat diet had about 60% of calories from fat. And we also have done studies with the Western diet with 34% of the calories is from fat. And what we found, we have similar findings with the Western diet. But what I wanted to demonstrate on this slide is that what we feel with one feeding a high-fat diet, there's results in gut microbial dysbiosis. And this actually through TLR4 can result in activation of the MAP kinase junk pathway, resulting in apoptosis and intestinal dysmotility. Also, what we showed in this study was if the mice are pre-treated for a couple of weeks with a prebiotic such as oligofructosaccharide, there is the changes that we see in the nitrogen neurons, as well as as seen here, is reverse, as well as the motility changes are also reversed. That is not shown on this slide. So what I want to demonstrate here is I showed you previously that lack of TLR4 was associated with a slow motility. But on the other hand, if there is excessive TLR4 stimulation, this also is associated with delayed gastrointestinal motility. So it's sort of like a bell-shaped curve. And then on this slide, I want to demonstrate one point which we find very interesting is the number of nitrogen neurons actually correlates with the less the number of nitrogen neurons, the slower the transit. And this was a study done by Francois Ricard in my lab. And he showed very nicely that as we lose nitrogen neurons, again, this was a high-fat diet, a Western diet model, the colon transit time, as you see on the left here, was significantly increased. And we use this as a model to, we feel that this validates our model of high-fat diet feeding. And so with this, I'm going to move on to our more recent studies. Recently, we published in JCI some evidence of a new cell death pathway that is happening when mice are fed a high-fat diet. And as you may know, pyroptosis is a sort of an inflammatory form of cell death in which pores are formed in the cell membrane, and it is dependent on caspase 1 or caspase 11. And I'm showing only one figure from this manuscript where we found that in caspase 11 knockout mice, there was, it could mitigate the effects of a Western diet, leading to a loss of nitrogen neurons. So again, we've demonstrated a couple of different ways of that cell death can occur, particularly neuronal cell death in these models. So having examined cell death through pyroptosis, we also wanted to understand what could be the potential mechanisms underlying the cell death, focusing again, as I mentioned at the beginning, on mitochondria. And before I get into the mitochondrial data, I want to talk a little bit about iron. Recently, there's been a lot of emphasis on iron in terms of iron metabolism, we know iron and its role in metabolism. We all know that iron plays an important role in several pathways in the cells, including mitochondrial function and oxidate and the electron transport chain. And I'm showing this article where recently published, where there's demonstration. They talk about the role of iron in neurodegenerative diseases, and particularly this phenomenon called ferroptosis. So the next couple of slides, before I finish, I'm going to talk about the some recent data from my lab on the role of iron leading to a neuronal ferroptosis and mitochondrial dysfunction. And I feel these pathways, these signaling pathways could be new targets for neurodegeneration within the ENS. So I'm just going to spend a minute on this slide just to talk about iron can be transported into the cell through DMT1, one of the iron transporters, or transferrin 1. Increased iron within the cell can contribute to the labile iron cell pool. Iron can come from mitochondria and contribute to the labile iron, as well as iron clusters can undergo autophagy, and the iron that's stored in ferritin can actually contribute to the labile iron pool. Now, I talk about labile iron. That's actually the toxic. If it's at high levels, it can be toxic. And the way it's toxic is actually what can, through a reaction called the Fenton reaction, basically ferrous iron can become ferric iron, and that happens actually in the presence of hydrogen peroxide, leading to formation of superoxide and high levels of oxidative stress. Now, the things that can mitigate this oxidative stress are GPX4, as shown on the slide, as well as glutathione. So GPX4 and glutathione can help prevent these reactions. In the presence of reactive oxygen species, there's membrane lipid peroxidation, and that ultimately leads to ferroptosis. So just to summarize, the things that what contributes to ferroptosis is high labile iron, the resulting in high levels of ferric, basically resulting in high levels of reactive oxygen species, reduced GPX4 and reduced glutathione, and ultimately membrane lipid peroxidation can lead to ferroptosis. And this is also shown on the next slide. Markers of ferroptosis as compared to other forms of cell death, there has to be evidence of lipid peroxidation, mitochondrial damage, certain gene expression changes, as well as increase in transferrin receptor 1, or also evidence of ferritin overload can all go along with ferroptosis. So what I'm going to show you is some recent data from my lab showing evidence of iron accumulation in enteric neurons. And this is work done by a postdoctoral fellow in my lab, Dr. Arun Balasubramanian. And as you can see here, these are enteric neuronal cultures, primary neuronal cultures from mice, and stained with different markers, PGP 9.5 as a neuronal marker, FTH1 is ferritin, and just DAPI to show the neurons, to show cells. And as you can see on the top, where under vehicle treatment conditions, we have very less ferritin expression. With treatment with palmitate, there is increased expression of ferritin. We are, as I mentioned, this is recent data. We are trying to localize exactly where the ferritin expression is increased. We see some co-localization with neurons, as well as some with other cells that we are trying to characterize. We do know that glia also store iron, so it's very possible that the ferritin is increased both in glia and neurons. As I will show you on this next slide, when we fed mice a high-fat diet for 12 weeks, as you can see, there is a significant co-localization of the neuronal marker TUJ1 and ferritin expression. So we do feel that with a high-fat diet feeding, there is increased ferritin expression, showing evidence of increased, iron stores. And I'm getting close. I know I'm getting close to the end of my time. I'm almost wrapping up here. Just to show on this slide, the one point I want to make is that with a high-fat diet, we believe, and we have not yet demonstrated this, that the inflammatory condition due to the high-fat diet leads to changes in the iron channels, which could be the reason for increased iron uptake in these neurons and increased ferritin accumulation. We also were able to show in enteric neurons, this is the enteric neuronal cell line, that there is the labile iron stained by ferronox, a dye to stain labile iron is increased in the presence of saturated fatty acids. And in the presence of chelators of iron, there is a reduction in this labile iron pool. Finally, the last two slides, I'm going to show you some evidence of mitochondrial dysfunction in the presence of various ways. When we manipulate the iron levels in neurons, we find evidence of mitochondrial dysfunction. And just to highlight on this slide, I'm going to show you just two parts. The vehicle curve as seen on the top here, and then when we treat with a compound called erastin or increase iron levels within these enteric neurons, there's a significant drop in the basically the bioenergetics in the mitochondria. And we are doing more studies to try to understand what saturated fatty acids as well as iron overload can do to enteric neurons. We also are studying mechanisms of what may be happening to the mitochondria, particularly looking at a process called mitophagy, which is basically when the mitochondria are taken up by the lysosomes and undergo basically in the process of mitophagy. And we feel that iron actually can affect this, iron overload can affect mitophagy. And what's shown on this slide is that in conditions of palmitate, here we're using a mitochondrial stain called mitotracker red. We see that there's abnormal accumulation when we treat with with palmitate or erastin, and we modulate and study mitophagy by treating with chloroquine. So these are all preliminary studies that we are pursuing further. I want to summarize by saying we believe the saturated fatty acid over time, within 9-12 weeks, we are now extending our studies to up to 6 months, over time can lead to neuronal loss. And this neuronal loss, we believe, is a mitochondrial dysfunction, plays an important role in this. And now we're studying the role of iron in leading to this loss. I've included one slide from Anita Vijay. She currently she works with Dr. Andrew Patterson, and one of the novel targets that she is studying is aryl hydrocarbon signaling. And I just wanted to highlight this one as another novel target for potential target in enteric neuronal degeneration. In the interest of time, I'm just going to mention that when she uses this compound TCDD, which induces AHR signaling, it leads to delayed intestinal transit. And she's doing further studies to understand how this happens. Just out of interest, this is a compound that's part of Agent Orange that many of you who work at the VA hospital may know that patients have been exposed to Agent Orange. And so I'm going to end, one second. Just by saying that today, I hope I've demonstrated to you that enteric neuronal degeneration can happen in various models, including diabetes and gut, and in models of high fat diet where there is dysbiosis, microbial dysbiosis, and there are various modes of dysfunction. We believe central to the neurodegeneration is the chronic inflammation from the high fat diet is a model that we can use to study ENS pathophysiology, and that is associated with mitochondrial dysfunction. And recently we are finding increased levels of iron overload. And with that, I will finish. And thank you for your time and looking forward to the discussion session.
Okay, thank you so much for that talk, which covered so much work in such a concise and clear way. Before I introduce our next speaker, I want to remind everyone of the format of this session, which is that we have a full 20 minutes at the end for questions. And the questions should be posted in the Q&A component of Zoom, but not in the chat. Okay, so it's my great pleasure to introduce our next speaker, Dr. Brian Gulbransen. Dr. Gulbransen is an MSU Foundation Professor in the Department of Physiology at Michigan State University, and he is also an ANMS counselor. He received his bachelor's degree in zoology and physiology from the University of Wyoming before completing his doctoral training in neuroscience at the University of Colorado Anschutz Medical Campus. He then completed his postdoctoral training at the Hotchkiss Brain Institute at the University of Calgary before joining the faculty at Michigan State University in 2012. Dr. Gulbransen's research focuses on mechanisms of neuroinflammation in the intestine, and he has a specific interest in the role of enteric glia in health and disease. Work from his lab has uncovered some of the fundamental rules that govern communication between glia, neurons, and immune cells in the gut. His research is currently supported by grants from the NIDDK, which focus on the roles of enteric glia in gut motility and pain. And he also has a program project grant from the NHLBI, and that focuses on interactions between adipocytes, nerve fibers in perivascular adipose tissue. The title of Dr. Gulbransen's talk is Intercellular Communication Between Enteric Glia, Immune Cells, and Neurons in Gastrointestinal Neuromuscular Disorders.
Okay, well, thank you for that introduction, Beth, and thank you, Shanthi, for the great introduction to this topic too. And I think this was nicely introduced, so I don't have to cover a lot of the background. And I'd also like to thank the ANMS for the opportunity to present some of our work on this topic. So, as many of you know, these disorders of neuromuscular communication are really a fundamental problem in the neural command center of the gut. And so this ability of the gut basically to run itself in the absence of input from the brain and spinal cord is because the gut has its own nervous system. And as Shanthi was telling you, this is the enteric nervous system, and most of this neuromuscular activity is regulated by these neural networks that sit between these two coats of smooth muscle. And the activity of the neurons and glia that reside in this plexus coordinate the contractions and relaxations of the smooth muscle coats to allow neuromuscular function to happen. So as you also learned in the last talk, this process is disrupted by inflammation, and inflammation is really a factor that links many of these disorders of neuromuscular dysfunction. And so this can cause motility to be delayed at sites of inflammation, to completely fail if the inflammation is severe enough. And what underlies some of these effects are changes in the contractions and relaxations, where there's a mismatch now in the contractions where the contractile component is enhanced and the relaxations are diminished. So the mechanisms that underlie neuromuscular dysfunction in disease include mechanisms that take place in the nervous system itself, altered immune responses, epithelial barrier dysfunction. And as you learned in the last talk, neuron death in the plexus contributes to some of this remodeling and some of the mismatch between the excitatory and inhibitory components. There's also synaptic facilitation and neuroplasticity in these remaining neurons. And so what I want to tell you today about is how these processes might happen, and they're not happening just in the neurons themselves, but they also involve interactions with the cells that surround these neurons, and these are the glia. And so these myenteric glia surround the neurons in these motor circuits, and they actually regulate the microenvironment of these neurons in health and disease. So normally these glia are functioning to support the neurons, regulate their activity, and to modulate the local immune environment. But in disease, these glia can actually contribute to killing off a portion of these neurons. And we found in previous work that activating these glia by purines during acute inflammation causes the death of some of these nitrogen neurons, and this contributes to some of the neuromuscular dysfunction following acute disease. So normally these glia and neurons are communicating to together in these motor circuits, and the neurons actually synapse onto the glia, and activating these synapses drives neuron to glia communication, and this can be appreciated in some of these videos. So this video is from a transgenic mouse that expresses a calcium reported in neurons in glia. The glia are co-labeled red here, and so you can see that activating the neurons with an electrical field stimulation drives fast activity in the neurons, followed by activity in all the surrounding glia. So activating glia has reciprocal effects on the neurons in the motor networks of the gut. And we've shown this several different ways. And one of those ways is by impairing the ability of the glia to signal to their surrounding cells. And so we did this by knocking out this membrane channel called connexin 43. That's a mechanism of gliotransmitter release from these cells. And mice lacking this gliotransmitter release mechanism show impaired colonic transit, and they also show impaired contractions in neuromuscular tension recordings. So on the flip side of that, you can also activate glia, and we used a transgenic model that incorporates chemogenetics to activate glia through engineered receptors called these chemogenetic HM3DQ receptors that couple to GQ signaling pathways and drive intracellular calcium responses in glia. And when you do that and study neuromuscular communication, we found that activating glia in this model was sufficient to drive neuromuscular contractions that were equal in magnitude as activating the muscle directly or activating the neurons. And we knew that this worked through neural networks because we could block the effect of activating glia with tetrodotoxin. So activating the glia increased colonic motility speed, increased the number of pellets produced per hour, and increased the propagation velocity of colonic migrating motor complex in organ bath studies. So some of these effects of the glia are because there are specific subpopulations of glia, and we recently found that how glia interact with the ascending and descending networks within these motor circuits actually underlies some of their excitatory effects on the motor networks themselves. So there's a large population of glia that's associated with the intrinsic primary afferent neurons, and then there are smaller subsets that are specifically activated by these descending pathways that would be these unipolar neurons projecting in the distal direction. And then there's another equally large population of glia, but non-overlapping, that's associated with these ascending pathways that leads to contraction. So this led us to this evolving model where there are specific subsets of glia devoted to these ascending and descending motor networks that control gastrointestinal motility. And the neurotransmitters that are involved in activating these glia include acetylcholine and ATP. And acetylcholine activity of the glia, or purinergic activation of the glia, tends to promote activation of the ascending motor network, whereas acetylcholine seems to act on the glia and have cross-inhibitory effects to refine some of the motor pathway responses. So if the glia are so important in these motor networks, we wondered if we might be able to find some evidence that there's a defect in glial signaling in a severe motility disorder. So to do this, we focused on a different transmitter system. And so this is the transmitter system driven by biolipids like lysophosphatidic acid. And so it's been known for about 20 years that glia are very responsive to this lipid signaling molecule. And they respond to it in cell culture. And actually, their responses in cell culture are larger than those to purines or to acetylcholine, as I showed in that previous slide. So the expression of the lysophosphatidc acid 1 is actually enriched in the glia, and this is confined to the glia in the myenteric plexus. So the neurons don't seem like they express this receptor, but it's very enriched in the glia. And we see this in the myenteric plexus of mice and the myenteric plexus of humans. So this is conserved from mice to people. So what's really nice about this is when you activate the glia with lysophosphatidc acid, the glia have these large calcium responses, and then this is followed by calcium responses in a subset of neurons that surround these glia. So then the functional effect of lysophosphatidc acid on gut motor function then is that it actually has a dose-dependent decrease in motility measured in organ bath studies. So this effect was observed in both the proximal colon and the distal colon, where increasing concentrations of lysophosphatidc acid actually decreased motility. So in contrast to that, you can block lysophosphatidc acid receptors with this drug called AM966. And we did these studies in vivo and then studied whole gut transit by measuring carmine red. And so, to our surprise, blocking lysophosphatidc acid 1 with AM966 also had an inhibitory effect on gastrointestinal transit and actually caused a severe failure of gut transit in a subset of mice that actually was fatal. So we think that this situation actually might mirror what happens during inflammation, because when we studied glial gene expression for LPA receptor 1 and the enzyme responsible for synthesizing LPA, these were both decreased in the DMBS model of colitis in glia. So based on these pieces of data, we speculated then that, that a loss of glial lysophosphatidc acid 1 receptor expression might occur in a severe motility disorder in people. So we looked in sections with from people with chronic intestinal pseudoobstruction. This was work we did in collaboration with Roberto DiGiorgio. And what we found is that normally there's glia surrounding neurons in these myenteric ganglia from people. And of course, the glia express very nice lysophosphatidc acid receptor 1. But this expression is lost in people with CIPO. So that led us to this hypothesis then that it's this loss of glial signaling function that might contribute to this severe motility disorder. So in contrast to that, if you actually sequence the glia during inflammation, it's actually many genes are upregulated. And so there's actually an overall gain of function instead of necessarily a loss of function. And many of the genes that are upregulated in glia during acute inflammation in mice are actually associated with immune responses. So many of these are linked to cytokine activity and immune processes. So we've begun investigating and starting to pick through different populations of immune cells that glia could be influencing that might alter the surrounding environment of the enteric nervous system and might contribute to neuromuscular function. And the first one I want to tell you about is this muscularis macrophage cell type. So why are we interested in muscularis macrophages? Well, one is because they're right next to glia and they're right next to neurons. And so this is one of these muscularis macrophages here labeled with MHC2. And as you can see, this macrophage is surrounding the enteric glia within this ganglion in a mouse. And this is some work from another group that used a reporter line to label some of these muscularis macrophages. And you can see it wrapping around some of the nerve fiber bundles in the myenteric plexus. And this same group actually used an antibody to block the CSF1 receptors, and that impairs the development of these macrophages. And when you impair their development, this actually leads to altered motility and neuromuscular muscle tension recordings. And this actually leads to more sporadic contractions. And then there's also some evidence from Gianrico Cipriani, who showed that changes in these muscularis macrophage populations contribute to dysmotility and disease. So it's a very interesting population of cells to study that has links with regulating motility, both through neuron interactions and then possibly through interactions with the glia. So we were really interested then when we started studying immune responses in DSS colitis, and we found that macrophage colony stimulating factor was increased during acute DSS colitis in mice. And so this data is quantified over here, and this is these are protein arrays from whole gut samples. So what was really interesting here was that this effect was lost in mice that lack this glial channel that I mentioned earlier that's responsible for gliotransmission, this connexin 43. So mice lacking the glial connexin 43 channels were unable to increase MCSF production during acute colitis, and that remained during chronic colitis. So when Vlado, who did this work, started looking at the cells that might be making this MCSF protein and isolating the cells using a membrane-bound MCSF antibody, because this membrane-bound isoform is the active signaling form of MCSF, found that many of these cells were not neurons or glia, but of those cells that were in the plexus that were labeled by Hugh or GFAP as neurons or glia, there were a lot of glia actually that expressed this. And in fact, more glia express MCSF, this membrane-bound form of MCSF than the neurons do. So glia are one of the major signaling hubs that's making this during acute DSS colitis. So the effect of this glial MCSF on the macrophages seems like it functions in regulating their activity. And so we showed this by measuring CD68 immunoreactivity as a marker of sort of a reactive macrophage phenotype, or a more pro-inflammatory phenotype of macrophages. And you can see an increase in CD68 immunoreactivity at the level of the myenteric plexus during chronic colitis, chronic DSS colitis in mice. And this effect is lost in mice that are unable to signal through the glial connexin 43 channels. Now, in this study, we were interested in visceral pain. So we were measuring visceromotor responses to colorectal distension. And we found that in these mice that lacked the glial connexin 43 channels, they were actually protected from developing this chronic visceral hypersensitivity following chronic DSS colitis. So it suggested that glial activity during acute DSS colitis or acute inflammation is actually responsible for making some of this MCSF that influences these muscularis macrophages to shift them toward a more pro-inflammatory phenotype. And that might impact some of these neuromuscular disorders in addition to pain. So I just want to finish by introducing another population of immune cells here that we've recently been looking at, and that's the neutrophils. And so we got into looking at these neutrophils actually kind of by chance, because we were interested in purinergic signaling. So this role of glia and neuroinflammation really is driven by high purine levels during acute colitis that act on these P2Y1 receptors. And so the role of glia in driving neuron death is actually driven by ATP and its metabolites acting through P2Y1 signaling pathways and then promoting ATP release from the glia. But as inflammation wanes, you have this breakdown in ATP and an increase in adenosine. And adenosine is usually thought of as an anti-inflammatory signaling molecule. Glia also express receptors for adenosine, and one of the main adenosine receptors that they express is this A2B receptor. And so the going thought was that these A2B receptors were anti-inflammatory, while the P2Y1 receptors were pro-inflammatory, and you might be able to capture different effects of the glia by stimulating one of these pathways over another. So we generated a new mouse model that lacked the A2B receptors in enteric glia. And so this was a tamoxifen-inducible knockout of the glial A2B receptors. And so you can see some immunohistochemistry images here showing that A2B receptor expression is lost in the glia in the transgenic mice, whereas it's very nice in the controls. And when we did this, we were actually really surprised about what happened to the cytokine responses, because we used acute DSS here again to study pro-inflammatory responses, and we found that these mice that lacked the glial A2B receptor signal, the ability to signal through A2B receptors, instead of having an increase in pro-inflammatory cytokines, they actually had a decrease in pro-inflammatory signaling cytokines. And one of these that really stuck out to us was CXCL1. And this was highly upregulated during the peak of colitis, and it stayed upregulated during the resolution phase of DSS colitis. And one of the others was IL-17 that was unchanged during the peak of colitis, but increased during the resolution phase of colitis. And both of these effects, both of CXCL1 and IL-17, were lost in these mice that were unable to signal through glial A2B receptors. So that suggested that glia might be making some of these cytokines like CXCL1. So we looked into the transcriptome data from glia during DMBS colitis, and we found that in DMBS colitis, sure enough, glia make a lot of CXCL1 mRNA. And then when we looked at glia in cell culture and challenged them with IL-1 beta, those glia actually made a lot of CXCL1. So there was about 1,000-fold increase in CXCL1 mRNA, and that was much greater than other pro-inflammatory cytokines that the glia were making. So the effect of this of CXCL1 production by the glia and of this glial A2B receptor knockout, we speculated that that might be on some of the immune cells then, because CXCL1 is a chemoattractant for some populations of immune cells. So when Vlado started to look into the populations of immune cells that were in the gut in these animals, he found no overall change in the percentage of CD45 positive cells, and many of these large populations like Ly6G positive cells, CD3 positive cells, and IL-17 positive cells were unchanged. But when he started to look a little bit more closely at subsets of these cells, then the changes started to emerge from this data. And the major change that he saw was that these cells that were double positive for Ly6G and IL-17 decreased in these mice that lacked glial A2B receptors. And the effect of losing this population of cells, which we think are neutrophils because of the Ly6G IL-17 double positive nature of these cells, the effect of losing this population actually protected these mice against persistent barrier dysfunction following colitis. So in wild-type mice, these mice exhibited increased permeability of the epithelium, both in vivo and in Using chamber experiments following acute DSS colitis. But these mice lacking glial A2B receptors were protected from this effect. And we think this is partially due to changes in the tight junction proteins expressed by the gut epithelial cells. Because in control mice treated with DSS, there's an increase in Claudin 1 and a decrease in Claudin 8. And these effects are lost in these mice that lack the glial A2B receptors. So we think that the glia in this context are actually, so this adenosine signaling through glia in this context is actually functioning to recruit some of these neutrophils, and then the neutrophils actually have detrimental effects on the gut by increasing barrier function. So I just want to finish with this summary then, kind of illustrating glia as this hub of signaling that's happening during these neuromuscular disorders, where glia can interact with the neurons to modulate enteric neuron activity and survival, and then through their interactions with neurons influence gut motility. Glia can also indirectly influence the neurons by influencing the immune environment surrounding neurons and at the gut epithelium, and they can influence their immune cell phenotype and activity. What I didn't tell you about today is some other work that we're doing focused on pain, where glia can contribute to this aspect of neuromuscular disorders by influencing the activity and sensitivity of visceral afferent nerve fibers that innervate the myenteric plexus. So I just want to finish by thanking everybody in the lab that does the work. We've got a great team. We are looking for new people, so if you're interested, give me a holler. And thank all my funding funding providers, and thank you for your support.
Thank you so much, Shanthi and Brian, for the outstanding talks. And we are entering now the Q&A phase of the webinar.
Tamas, do you have questions to start out with? As people, I understand what this is like. You have to type the whole question in, and it can take a while to get it all in there. So sometimes there's a little bit of a lag. I certainly have a couple of questions, but if you would like to go ahead.
Yeah, certainly. We would like to encourage the audience to ask questions. Currently, the box is still open, so please do enter your questions into the Q&A box. In the meantime, let's start the questions from the. Oh no, there is
Oh,
one.
we got a question.
All right, so this is from Dr. Sharkey. Hi, Brian, great talk. The glia involved in barrier function are distinct from those of the enteric glia that you showed us data from. Any thoughts on this?
Yeah, it's a great question. And in this model, we're not able to differentiate between the effects of the two because we're using a SOX10 driver to knock out these glial A2B receptors. So in this model, we're really losing the A2B receptors in all the enteric glia. And we don't know necessarily if this is a specific effect by these mucosal glia or from some effect of the myenteric glia or a combination of all of these effects. It could be any combination of those at this point. It definitely is possible that there are some local signaling things going on in the mucosa, and most of the effects of this model were actually focused on the mucosa. We actually didn't see a lot of effects on motor function, overall inflammation, or changes in the myenteric plexus or overall disease severity. So it seems like most of the activity in this model anyway is focused on the mucosa. But it's a great question because there may be some differences in how these different glia and the signaling contributes to different aspects of the neuromuscular function.
Okay. Our next question comes from Dr. Henry Parkman, and it's directed to Shanthi. Does iron overload have a role in some of the GI motility disorders, such as chronic constipation, gastroparesis, or achalasia?
So that's an excellent question, Dr. Parkman. So we are actually, just recently only we have started investigating the role of iron. And we do think that it may have a role in all the different conditions you mentioned, gastroparesis, although I don't have right now any evidence for that. We're kind of, what we feel is in conditions of chronic inflammation, there is increased iron overload, and that then may contribute to these neuronal changes. So definitely a good thought and something that we are thinking of investigating further.
Thank you. I have a follow-up question actually to this. Do you think there is any role for extramitochondrial iron handling mechanisms like the cytosolic iron sulfur assembly in the iron overload induced neurodegeneration?
Yes, I think so. We're trying to understand it, and we think it's both the cytosolic iron as well as it's possible the mitochondrial iron levels are high. So I think both cytosolic and mitochondrial can contribute to oxidative stress and thereby neuronal degeneration. So definitely the And did you mention, Tamas, extracellular iron? Is that
extra mitochondria.
extra mitochondria? Yes, very good. Thanks.
I actually have a follow-up to Dr. Sharkey's question as well for Brian. It's related, and I guess I'm interested in the epithelial cells because I believe, isn't CXCL1 the receptor for IL-8? I think it is. And so I know you spoke about communication with a lot of different cell types in the area, and that made me wonder about epithelial cells and whether there is a role for the glial epithelial.
Yeah, I mean, that's kind of the open question right now too, how glia could influence or interact with the epithelial cells themselves. We had some evidence from the DREAD model actually that stimulating the glia, you couldn't block all the effects of stimulating the glia on the secretomotor function with tetrodotoxin. So it suggested that there may be some of the residual effect could be from direct interactions with the epithelium. And then Michelle Neuenlist has this whole literature of glia influencing all these different aspects of the epithelium. So epithelial cell maturation, migration, differentiation, things like that. So there's definitely the possibility that they're interacting with the epithelial cells. And I think the cell culture data on that's really strong. How they do that in vivo is more questionable about how that process really works.
Thank you.
Thank you. Now I have one more question for Shanthi. So the mitochondrial dysfunction is likely universal, affecting all neurons. Why do you think nitrogen neurons are particularly sensitive to that?
Yes, yes. It's an excellent question, Tamas. And what we think is maybe the contribution of nitrogic stress. So the NO itself may somehow, with the iron overload, and then, so we haven't teased out the mechanism, but that's something we are trying to figure out, and whether the actual NO release may be contributing to this susceptibility for the nitrogic neurons. And definitely we need to understand whether there's differences in mitochondrial sensitivity or, dysfunction in nitrogic neurons versus cholinergic. So something that we're examining and trying to understand. But thank you for that. Mm-hmm.
I also have a question for you, but I would like to say that please, we encourage people to put questions. I've been in your shoes and not wanted to type the whole thing. I will not read your typos. Well, we will read the question as it's I understand you have to type it all in instead of just saying it, but please, we really welcome your questions. But I will ask a question, which is that CAS1, I feel that I've read about in relation to age-related neurodegeneration, and that it has a strong role in that. And I'm wondering if you're looking at all at how aging may mediate what you're finding.
Yes, it's an excellent point. And so far, so we have particularly haven't studied aging, but you're absolutely right we believe with in enteric neurons in older mice using the mouse model. Right now we're going up to 6 months, both wild type and high fat diet fed mice, and we are going to study the effects of age in that model. So definitely, and I think similar mechanisms can contribute to neurodegeneration in that setting. Thank you.
Thank you very much. I'm afraid this is all the time that we have. Should there be any further questions, please still enter them into the Q&A as long as it is open, and those will be answered on Doc Matter. And with that, I'd like to thank the speakers again for the wonderful talks, and I'd like to thank the audience for their interest and the participation. Thank you.